everyone, and welcome to today's session. I'm excited to introduce our topic, Study Session 2, Generation of ATP in Glycolysis Pathway. Today, we'll explore how cells produce energy through glycolysis, focusing on the process of ATP generation. We'll go over the key steps involved and understand how this pathway fuels our cells. Let's get started. Welcome to our presentation. In this slide, we're outlining the structure of our discussion. First, we'll start with the introduction to set the stage for understanding glycolysis. Then, we'll look at the learning outcomes so you know what to expect and what you'll be able to do after this session. Moving on, we'll delve into the main content, which covers how ATP is consumed and generated during glycolysis, an essential energy pathway. We'll also examine how this process is regulated at different points, ensuring efficient energy production. Additionally, we'll explore some clinical conditions that arise when glycolysis is impaired, highlighting its importance in health and disease. After that, there will be tutor-marked assignments, either individually or in groups, to reinforce your understanding. We'll conclude with a summary of key points and then provide self-assessment questions and answers to test your knowledge. Finally, we'll share references and further readings for those interested in exploring the topic more deeply. This overview will help guide us through the detailed concepts ahead, connecting each part to the bigger picture of cellular metabolism. In this session, we continue our discussion of the glycolytic pathway. As you may recall, during this process, 2 ATP molecules are produced. However, it's important to note that even more ATP is generated when pyruvate is fully oxidized to carbon dioxide and water in the mitochondria. The regulation of glycolysis mainly involves 3 key enzymes that catalyze its irreversible reactions. Among these, the most critical control point is the enzyme phosphofructokinase, or PFK, because it catalyzes the first irreversible step that is unique to glycolysis. Now, once pyruvate is formed, it has 3 possible fates. It can be converted into acetyl-CoA, ethanol, or lactate, depending on the cellular conditions. Understanding these pathways is crucial because impaired glycolysis can lead to clinical conditions such as lactic acidosis or pyruvate kinase deficiency, which highlight how vital this process is for normal metabolism and health. After studying this study session, I expect you to be able to understand the gross and net ATP yield of glycolysis, which is fundamental for grasping how energy is produced in cells. We'll also go over the key glycolytic regulatory enzymes and their effectors, helping you see how this pathway is tightly controlled. Additionally, I want you to be able to explain the different fates of pyruvate, whether it enters the mitochondria for further oxidation or is converted into other compounds. We'll also cover some disorders of the glycolytic pathway, so you can appreciate how disruptions can lead to metabolic issues. Overall, these learning outcomes will give you a solid understanding of glycolysis, its regulation, and its clinical relevance. Let's take a closer look at how ATP is both consumed and produced during glycolysis. As you can see in this table, the process results in a net gain of 2 ATP molecules. It's important to understand that while some ATP is used up early on, the overall process generates more energy than it consumes. Most of the energy we extract from glycolysis isn't stored there. Instead, it's harvested later in the TCA cycle and the electron transport chain. These steps are where the real energy payoff happens, allowing our cells to produce the majority of ATP needed for various functions. So this table helps us see the balance, how initial investment in ATP sets the stage for a larger energy return in subsequent stages. It's a key part of understanding how our cells efficiently extract energy from glucose, fitting into the bigger picture of cellular respiration. The glycolytic pathway has a dual role in the cell. First, it degrades glucose to generate ATP, which is the energy currency the cell needs for various functions. Second, it provides building blocks for synthetic reactions, supporting the cell's growth and maintenance. The rate at which glucose is converted into pyruvate is carefully regulated to meet these 2 major cellular needs. If the cell requires more energy or building blocks, the pathway can be adjusted accordingly. Now, when we think about regulation, enzymes that catalyze essentially irreversible reactions are key control points. These reactions act as major gateways, so to speak, and their enzymes are prime targets for regulation. Specifically, in glycolysis, the reactions catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase are virtually irreversible. Because of this, these enzymes don't just facilitate the reactions, they also serve important regulatory roles. Understanding regulation in glycolysis helps us see how the cell balances energy production with biosynthesis, ensuring everything runs smoothly according to its needs. Now let's talk about PFK, which stands out as the most important control point in glycolysis. This enzyme is tightly regulated because it essentially decides whether the pathway speeds up or slows down. Interestingly, ATP, our cell's main energy currency, actually inhibits PFK when its levels are high. It binds to a specific regulatory site, which is different from the active site where the reaction occurs. This means that when there's plenty of ATP, PFK activity decreases, slowing down glycolysis. However, this inhibition isn't permanent. When AMP levels rise, indicating lower energy availability, it reverses this inhibition and activates PFK, increasing the pathway's activity. So, the enzyme's activity is really sensitive to the ATP to AMP ratio. Lower ratios signal energy scarcity, prompting more glycolysis. Another key regulator is citrate. When citrate levels are high, it suggests that the cell has enough biosynthetic precursors. Citrate further inhibits PFK by amplifying the inhibitory effect of ATP, helping the cell avoid unnecessary energy production and precursor synthesis. It's also worth noting that enzymes like hexokinase and pyruvate kinase play roles in regulating glycolysis, especially at the committed step, the first irreversible reaction unique to this pathway. This step's regulation is crucial because it commits the substrate to glycolysis. Overall, PFK is most active when the cell needs both energy and building blocks. When energy or carbon skeletons are needed, the enzyme ramps up activity. But when both are abundant, PFK activity is almost shut down, helping the cell conserve resources. So, regulation of glycolysis, especially through PFK, ensures the cell responds appropriately to its metabolic demands. Now, let's take a closer look at how hexokinase and pyruvate kinase are involved in regulating the rate of glycolysis. Pyruvate kinase, specifically from muscle and liver, is allosterically inhibited by ATP. This means that when the cell has a high energy charge, or plenty of ATP, the enzyme's activity is reduced, effectively blocking the conversion of phosphoenolpyruvate, or PEP, to pyruvate. This feedback helps prevent unnecessary energy expenditure. Similarly, hexokinase is also subject to allosteric regulation, but in this case, it's inhibited by glucose 6-phosphate. When G6P levels rise, often due to a slowdown downstream, hexokinase activity decreases, preventing excess accumulation of G6P. Interestingly, when phosphofructokinase or PFK is inhibited, levels of fructose 6-phosphate, F6P, increase. Since G6P and F6P are in equilibrium, this leads to an increase in G6P as well. So, in essence, inhibition of PFK not only slows down glycolysis at that step, but also causes a rise in G6P, which then inhibits hexokinase. This interconnected regulation ensures a balanced control over glycolytic flux, preventing wasteful overproduction and conserving cellular resources. This coordination highlights how regulation at multiple points maintains overall metabolic homeostasis. The fate of pyruvate in generating metabolic energy actually varies quite a bit depending on the organism and the specific cell type. It's fascinating because pyruvate can be channeled into different pathways, leading to different end products. For example, in some microorganisms like yeast, pyruvate is converted into ethanol through a two-step process. This is a classic example of fermentation, especially under anaerobic conditions. In contrast, in muscle cells during intense exercise, pyruvate is often reduced to lactate when oxygen is scarce. And in most other cells, pyruvate is transformed into acetyl-CoA, which then enters the citric acid cycle to produce energy efficiently. So, understanding these different pathways helps us see how organisms adapt their metabolism to their environment and energy needs. A lot of energy in our bodies is produced aerobically, meaning in the presence of oxygen, primarily through the TCA cycle and the electron transport chain. Now, the starting point for this oxidative pathway is a molecule called acetyl coenzyme A, or acetyl-CoA for short. This molecule is crucial because it feeds into the cycle, enabling us to generate energy efficiently. Acetyl-CoA itself is formed inside the mitochondria, which are often called the powerhouses of the cell. The formation occurs through a process known as oxidative decarboxylation of pyruvate. So, pyruvate, which is a 3-carbon molecule, reacts with NAD+ and CoA to produce acetyl-CoA, along with carbon dioxide and NADH. This reaction is pretty important because it links glycolysis, where glucose is broken down into pyruvate, with the TCA cycle, allowing energy to flow smoothly through our metabolic pathways. The entire process is catalyzed by a complex enzyme system called the pyruvate dehydrogenase complex. Understanding this step is key because it highlights how our cells convert food into usable energy, especially in aerobic conditions. It connects the initial breakdown of glucose to the energy-producing steps that follow in the mitochondria, tying together the overall theme of cellular respiration and efficient energy production. This is the most common form of metabolic acidosis that we encounter. It typically happens when there's an overproduction of lactate, or sometimes when the body isn't using lactate efficiently. It can also occur if there's inhibition of pyruvate dehydrogenase, which is a key enzyme in energy metabolism. Additionally, this type of acidosis can be caused by rare congenital disorders where mitochondria fail to function properly, or in conditions like diabetic ketoacidosis. Liver or kidney disease can also contribute to this problem. Clinically, we see that lactate levels are greater than 5 millimoles per liter, and the serum pH drops below 7.35. Patients often present with symptoms such as nausea, vomiting, hyperventilation, which is the body's way of trying to compensate, and sometimes irregular heart rates. In essence, this form of lactic acidosis is closely linked to impaired glycolysis and energy metabolism, highlighting how disruptions in these pathways can lead to significant clinical consequences. Understanding these connections is crucial as we look at the broader picture of metabolic disturbances. So, here we're looking at a rare genetic defect in glycolysis that specifically causes hemolytic anemia. What happens in this case is that intermediates in the glycolytic pathway, especially those close to the pyruvate kinase step, tend to accumulate. At the same time, you'll see a decrease in pyruvate and lactate levels. This imbalance impacts the red blood cells' energy production, making them more prone to destruction. This increased destruction, or lysis, of red blood cells can lead to jaundice, which is due to the buildup of bilirubin from the breakdown of hemoglobin. The key point here is pyruvate kinase deficiency, which disrupts normal glycolysis in red blood cells, leading to their premature destruction and resulting in hemolytic anemia. This highlights how a single enzyme defect can have significant effects on cell survival and overall health. Let's wrap up our discussion on glycolysis by focusing on how ATP is generated during this process. Throughout this session, our main goal has been to deepen your understanding of the glycolytic pathway and its significance in cellular metabolism. Remember, glycolysis involves both the consumption and production of ATP. Initially, ATP is used to activate glucose, but as the pathway progresses, it produces ATP through substrate-level phosphorylation. This net gain of ATP is crucial because it provides immediate energy for the cell, especially in anaerobic conditions. We also explored how glycolysis is tightly regulated. Key enzymes like hexokinase, phosphofructokinase, and pyruvate kinase act as control points, ensuring the pathway responds efficiently to the cell's energy needs. This regulation is vital for maintaining metabolic balance and adapting to different physiological states. Furthermore, we discussed clinical conditions related to impaired glycolysis. For example, deficiencies in enzymes like pyruvate kinase can lead to hemolytic anemia, highlighting how crucial this pathway is for normal cellular function and overall health. To sum up, understanding the mechanisms of ATP generation, regulation, and associated disorders not only enhances your grasp of glycolysis, but also underscores its importance in health and disease. Keep these concepts in mind as they form the foundation for further studies in metabolism. Let's go over some key review questions. First, what is the gross and net ATP yield of glycolysis? The gross yield is 4 ATP molecules per glucose, but because 2 ATP are used up front in the process, the net gain is 2 ATP molecules. This makes glycolysis an efficient way for cells to quickly generate energy. Next, let's look at the glycolytic regulatory enzymes. These are crucial control points, including hexokinase or glucokinase, which catalyze the first step, phosphofructokinase 1, a major rate-limiting enzyme, and pyruvate kinase, which catalyzes the final step. These enzymes help regulate glycolysis based on the cell's energy needs. Moving on, what happens to pyruvate after glycolysis? Pyruvate can follow several paths. It can be converted into lactate under anaerobic conditions, which is important in muscles during intense activity. Alternatively, in the presence of oxygen, pyruvate enters the mitochondria and is transformed into acetyl-CoA to fuel the citric acid cycle, producing more energy. Finally, let's discuss some disorders of the glycolytic pathway. These can include enzyme deficiencies like pyruvate kinase deficiency, which leads to hemolytic anemia, or phosphofructokinase deficiency, causing exercise intolerance. Understanding these disorders highlights how vital proper glycolytic function is for overall health. So these questions help us connect the biochemical pathways to their physiological importance and clinical relevance, reinforcing how glycolysis fits into the bigger picture of cellular energy metabolism.